All right, let's talk about potential. Your collective potential as a group of young budding anatomists. More specifically, membrane potential. Membrane potential. We all have. We'll talk about the resting membrane potential. We'll talk about how that deviates from normal. Graded and action potentials are ways that we can deviate from our resting membrane potential. And every cell in the body, well, every excitable cell in the body has a resting membrane potential that it uses to achieve some work of some kind. We're going to see a really nice descriptive example, nice video of membrane potential today. I found these and I put them in the notes that you have on Moodle. And if I were to click on one of these links, it would take you to the Wiley Plus assets with a couple of different videos, 6 or 7 minutes worth of 3D rendered video. And it's a really nice way to bring all the lecture components together. So I encourage you to go through them. We're going to watch the second one today after we set the stage for what membrane potential is and why it's generated. Then we'll talk about how it's affected by graded potentials to make action potentials. Really useful videos here if you're bored of listening to me talk or reading your textbook. So potential as the baseline allows us to deviate from that baseline. And deviate means communicate. It allows us to communicate between excitable cells in the body. When we deviate from rest, the magnitude of that deviation or how frequently we deviate creates an impulse, and that impulse means something to tissues in the body. And meaning something is the basis for communication. So if we have a resting potential and we can change it in an ordered fashion, then we can communicate from one end of the body to another. And that communication is largely done over large distances by action potentials. So between the nervous system and an effector tissue, an action potential is the signal that goes between those two areas. That's pretty far when we think of cellular terms. In terms of the body, that's pretty far. The signal can travel that far. It can also travel over short distances, depending on the length of the nerve, but when we think short, we often think of contained within one neuron, or maybe between neighboring neurons, but certainly contained within one neuron. Communication at that level is usually done through graded potentials, deviations of what we exhibit at rest. Short distances here is within the same cell, and that's what we're going to use as our understanding moving forward. So I'm just checking to make sure that it's not cut off. I made the screen or the slides a lot wider, so you should be able to have more real estate to see everything. Now, graded potential, action potential is the deviation from normal. So we have to assume that there is some baseline. We need there to be a pre-existing state for a graded potential to affect that resting state. And that resting state is the resting membrane potential. And so we're going to discuss today how that membrane potential is generated, what it is by definition, and how we can use it as the basis for signals in graded and action potentials shown here. I'll say the word potential a lot today. And by potential, usually what I mean is the cell is polarized, or whatever endpoints that I'm looking at are polarized. At rest, that potential is evident because most cells are polarized. And simply what I mean by polarized is on two ends there are opposite charges. So think of a magnet that you had as a kid. It had a north pole and a south pole, or a positive pole and a negative pole. Those were different charges. And it was polarized because they had different charges on each end. We aren't thinking in terms of magnets, though. This might be the most accessible example, but in the body, the cell isn't necessarily a stick with two different ends of different charges. It's polarized in another way. The cell is polarized from the inside out. So if I take my magnet and expand it a little bit, so that this box, this cartoon box, is a representative cell. The inside is the cytoplasm. The line around the outside is the plasma membrane, indicated by the PM. And when I say polarized, this is what we mean in biological terms. The inside has a more negative charge relative to the outside. Same idea as the magnet. There's a difference in charge. But here it's from the inside to the outside, not end to end. It's across a membrane. And this is the basis for the resting membrane potential, and if we can ever affect one or both of those charges, we create a graded or action potential. In the resting situation, with a polarized cell membrane, this is what we're talking about. This is largely due, and we'll see why, to a difference in the distribution of cations, positively charged ions. There's more to it, but the initial major difference between the inside and the outside of cells is in the distribution of sodium and potassium. So inside the cell we have a lot of potassium, very little sodium, and the opposite is true outside. And we want to maintain that. We have countermeasures in place to make sure that that always stays the same, or that is the case. So if we are trying to communicate between different areas of the body and send a signal and affect potential, the charge, we do that by affecting the concentration of these ions, or the localization of these ions. If we can move them, we can affect the charge. If we can move them, we can create a signal. If we can move them, we can communicate. So this is important to understand as we progress through our discussion, the division of cations. Sodium, high outside, low inside. Potassium, the opposite. High inside, low outside. Now it's not immediately clear why the division of positively charged ions alone will cause a positive and negative differential, or a differential in charge. They're both positive, but that will become more clear as we go through. That's better, not so bright now. So from this resting situation, I guess three things can happen. One, membrane potential can stay the same. We can maintain the difference in charge across the membrane. In practice, that doesn't happen. Even when no signals are being sent, the potential always varies a little bit. If it varies upwards, if the cell becomes less, this is always going to be confusing. You've got to think about how you're going to present this first before you say it. If the potential increases, becomes less negative, then the cell is said to be depolarized, or moving towards being depolarized. The opposite is true. If the potential becomes more negative, it's hyperpolarized. And this is relative to whatever our resting situation is. They can depolarize. In that fully depolarized state, then the positive and negative compartments that I showed you would be equal. Hyperpolarized is simply the inside is more negative than it was at rest. And we summarize it with a nice little cartoon plus and minus sign. We think of the entire inside of the cell as being negative, or the entire outside as being positive. But there are many individual contributors to that net charge. And we're not going to study too many of them. We'll talk about a few of the main factors. But it's the distribution mainly of those positive ions we talked about. The negative ions aren't so much of a concern because they don't move as much as the positive ions do. And the reason for that is the molecules, the pieces that are negatively charged, are larger, and they tend to stay inside the cell a lot more easily. They're attached to phosphates, PO4 negatives, lots of atoms, big atoms that keep the molecule inside, or they're part of proteins. They don't move through the membranes easily. So it's largely the distribution of ions, mainly the ones that we talked about. So let's consider, in terms that we already know, the sending of a signal. The sending of a signal. This is going to be very similar to the reflex arcs that we talked about, but it's not a reflex. This is a sensory signal that is processed to create an action plan and move the hand. And the example in the book goes as follows. Those of you that have pens or pencils can follow along, or you can substitute in as a surrogate a keyboard. The idea that your hand is touching something and you know how it feels. So if I sense something in my hand, I have pressure sensors in the tips of my fingers. We talked about that in the lab already. And those receptors themselves are not nerve endings per se, but they are closely associated with nerves. They're a special kind of receptor that we'll talk about how they're activated shortly. But those receptors respond to light, pressure, and touch. And those receptors generate a graded potential. They change from their resting situation. The potential changes from its resting situation when my fingers are in contact with keyboard, pen, pencil, or laser pointer. Which is good. So I want that graded potential, I want the membrane potential to change so that I know I'm in contact with something. That information is relayed. We've talked about the relays at length. The graded potential in the receptor is focused on a nearby sensory nerve, which causes a signal within that nerve. That signal is relayed on the sensory side, indicated by the green arrow here, until we reach the integration center, until we meet up with an interneuron. Here it's in the brain, in the central nervous system. Notice this afferent signal goes right to the posterior column of the spinal cord, which we know are somatic. They're bringing to your conscious awareness that type of sensory information that's being relayed, that touch information that's being relayed. So right to the posterior column, I'm up in the brain where the afferent synapses with an interneuron, and there's a series of cascading synapses, and that's the integration of the signal, the processing of the signal. Only when that cascade reaches the cortex am I aware that I'm holding onto this laser pointer? Or are you aware that you're touching a pencil or the keyboard? The potential generated in the touch receptors traveled all that way to your cortex. Now you're aware that this is happening. If you were paying attention last class and you're looking at this example, you think, oh, the thumb and the lateral two fingers are what are sensing touch. And what nerve would probably relay that sense of touch to the brain? Median nerve of the brachial plexus, right? If you were paying attention. So we have these cascading series of synapses in the brain. How they activate or in what order they activate is not important now. We'll talk about the order later. But that they activate and bring this to our awareness, our conscious awareness, is important. Now, if we're trying to write, or if I'm trying to point at the screen with my laser pointer, I execute a motor plan in that same area. I have sensory information on how much pressure is being added by my hand to the laser pointer, and I activate muscles in my hand. I send a signal from what we call an upper motor efferent to a lower motor efferent, and that signal goes towards the muscles of the hand. I was able to take that signal generated from light, touch, and pressure, communicate that information to the cortex where I'm aware that I'm touching something in my hand, and then achieve some downstream action. Here writing or pointing the laser pointer at the screen in a coordinated manner. And this is an oversimplified diagram. We have one series of afferent nerves, one series of efferent nerves going to all the muscles of the hand that allow you to write. It's not as simple as that, I would say. This entire relay started with a graded potential and is supported by the movement of action potentials through the entire loop, the entire relay. So let's talk about the generation of potential and the propagation of that signal. A lot of it has to do with the fact that in excitable cells, where sodium and potassium are divided and separated by the plasma membrane, we have the ability to move those ions across the membrane through ion channels. These are specific conduits for ions of interest. They usually only work for one type of ion, not another. They're too small for big items to move through. And they're activated in response to a certain stimulus. We have four main types of ion channels. We'll talk about each of those in turn. They can open and close, as is indicated by the gate. So you have the potential to open them if you want to, or if the appropriate signal is present. And if they're open, there's no guarantee that sodium or potassium or any other ion will move. There's no guarantee just because it's open that things will move, things will happen. Once this gate is open, say it's a sodium gate. Once this gate is open, two things drive or decide whether or not sodium is going to move. The electrical gradient and the chemical gradient. What do I mean by that? Electrical gradient. Positive is attracted to negative. If I'm sodium, I have a positive charge. I live outside of the cell. If my gate opens, there's a negative charge inside the cell. Immediately I want to move in. That's one factor. Second factor, chemical gradient. High concentration to low concentration. I'm sodium, I'm outside the cell, I have a lot of friends nearby. There are very few inside the cell. crowded out here. I want to move in. Easiest example. A somewhat harder example is that of potassium. And we can see that here. This is one type of channel that we have available. This is called a leak channel, which is gated, but is usually open. It makes the membrane leaky, which essentially allows passive movement of these ions across. Not in a massive rush, but in a small trickle. In this example, we have potassium, which we know lives inside the cell, shown down at the bottom, the orange or peachy colored triangles, wanting to leave through the leak channel to go into the extracellular space, outside of the cell. So if I think back to the example I just gave you with sodium, the two things that drive or dictate whether I want to move as a potassium ion now are the electrical gradient and the chemical gradient. Well, if I'm inside the cell and I'm potassium, there's a lot of friends, a lot of other potassiums nearby, few outside, so I want to leave. But how is the charge? How would you describe the charge? If you're potassium looking outwards, what is the gradient, the electrical gradient? Does it influence you to move? First of all, okay, who wants to venture a guess? What charge is potassium? Positive, okay, +1, positive. So I am attracted to what charge? Negative charge. Where do we find negative charge? Inside the cell. So I have conflicting gradients. There's less opportunity. There's less stimulus to move out of the cell. The charge wants to keep me in. The concentration wants to push me out. It ends up that you leave the cell. As a potassium ion, you leave. You go against the electrical gradient because the chemical gradient is so large and so forceful. So this leak channel typifies that idea. You see potassium leaving. There are leak channels for sodium as well. And a leak channel for sodium would allow it to leak in. They would move in the opposite direction. More sodium outside than in. It so happens that there are more potassium leak channels than sodium leak channels. And that's one important factor in creating membrane potential. And while this says they alternate between open and closed, they do, which is what allows for a trickle of flow. But they tend to do it pretty regularly, and we'll leave it at that. They randomly open and close. They allow leakage of ions through the membrane pretty regularly. So more potassium than sodium, that means there's more likelihood potassium will move out. We'll come back to that when we talk about net charge. Second type of channel, ion channel in neurons or other excitable cells, are what we call ligand-gated channels. And ligand is simply referring to a molecule of some kind. Something binds to this channel. This channel is not open unless that thing is bound. It's not leaky, it's not intermittent. It needs to have a ligand present in order for it to open. And the best example that we have, at least when we talk about action potentials, is the acetylcholine ligand-gated channel. We talked about acetylcholine already. Yesterday, briefly, that's a neurotransmitter that relays the nervous signal between nerves and muscle cells. Well, this purple channel, it's a conduit that will allow ions to move through, like another ion channel, but it's closed on the left because acetylcholine, this red triangle now, hasn't bound yet. When it does bind, as is the case on the right-hand side, it opens up. and here allows relatively free movement of ions. Sodium and calcium are allowed to move in pretty easily in this case. I think potassium even moves out. This is a fairly large channel. It's not as specific. Not as specific, it's larger, which allows for greater flow. And a greater flow will disrupt the membrane potential more easily and more severely. Which might be important for relaying nervous signals. Third type of ion channel, mechanically gated ion channel. Mechanically gated. To open these channels, you physically pull them apart. Their structure is physically deformed. This can be from deformation of the plasma membrane, as is the case with touch receptors. So holding on to my laser pointer, generating a potential, was mostly through opening of mechanically gated ion channels. But it could be any other physical stimulus too. So pressure, touch is one. Sound waves can open mechanically gated channels in the ears. Auditory signals are generated from sound waves opening, or in part from sound waves opening these channels. So there's some physical change in the structure that you can see opens up the top piece which blocks the channel normally. The lid is removed from the can, so to speak, and ions can move through. And lastly, the channel that's really responsible for the propagation of nervous signals, the voltage-gated, I want to say sodium channel because that's the main one that we always talk about, but a voltage-gated ion channel. This one happens to be potassium. Call me a liar. A voltage-gated channel doesn't respond to physical stress, doesn't need a ligand to bind, isn't leaky. It only opens when we reach a threshold membrane potential. So whatever it is at rest, if it increases, becomes more positive, and depolarizes, those are all the same thing. If it becomes more positive and we reach a point that is specific to each kind of voltage-gated channel, we call it a threshold, then these will open up. And this is very important when we get to depolarization and the chain reaction of propagating an action potential down an axon. The disturbance moves in a wave, and these open in a wave as well as you move down and propagate that signal. So these are the types of channels. We are going to put them into their respective roles when we actually discuss in detail the creation of a potential. But to do that, we need to start with the baseline scenario, the resting scenario. We're going to define the resting membrane potential, the charge and the value. We'll talk about the main characteristics that cause the resting membrane potential, and then we'll talk about how it's disturbed. So we already know that it's polarized. The cell is polarized from the inside out. The inside is relatively more negative than the outside. This is a normal resting scenario, and we create it by three main stimuli, or there are three main causes for this negative differential. So there are an unequal distribution of sodium and potassium ions. That's one. We've established that. Being that there are more potassium leak channels than sodium leak channels, that means that more potassium will move out than sodium will move in. Which, no matter what the stoichiometry, if more positive charge is moving out than moving in, the inside becomes more negative. We also actively create this differential, and that's number 3, but we'll get there in a second. So you can see that shown here in this inset figure. The differential in charge across the membrane generally only exists immediately in the vicinity of the membrane, along the surfaces, but it's still there. Notice along the top surface there is an abundance of positive charge. Along the inner surface there's an abundance of negative charge. And as you move away from the membrane, regardless of which direction, it ends up starting to balance out. We don't care about the balanced portion though. We care about the differential at the membrane. So we have less positive charge on the underside, more positive charge on the top side. There is a difference in charge across this membrane. More potassium moves out than sodium moves in, so more plus leaves the cell. Second factor that helps to generate this resting potential is that you would expect negatively charged things to move to destroy this difference, to destroy the differential. They'd move from the inside to the outside on top of the membrane because they're being pulled. Negative is attracted to positive, positive to negative. There is a charge or an attraction for negative things to move from the inside to the outside to satisfy or neutralize this charge. But the anions can't move out. And we actually see it in a little bit more detail coming up in 2 slides. But for the same reasons that I mentioned already, the anions, the negative particles, are big. They're small organic molecules or large clusters of atoms. They're big. They can't move out. So they can't dissipate this differential. And lastly, sodium-potassium ATPases. These are energy-consuming pumps that work to create this gradient, to create the differential of sodium and potassium ions. So the sodium-potassium pumps are always active. They're always consuming energy, even sitting here at rest. They're a big part of your resting metabolic rate. And their job is simply to move sodium out of the cell and potassium back in. Which is good. That's the normal situation. We want that to occur. For normal cell function, we want that disparity. If there's ever sodium in the cell, these will pump it out. And to do that, at the same time, they move potassium back in. The problem is, it's an unequal exchange. 3 sodiums move out for every 2 potassium that move in. Meaning you lose 1 charge for every pumping of this pump. And it happens, there are thousands of pumps in every cell. It happened right there for all of you, and again, and again, and again. It's always happening. It's a constitutive process. So how do we measure potential? I've said negative charge inside, positive charge outside. I've also said less positive inside and more positive outside, and they're both correct. The potential is just a difference in charge. A difference in charge. and it literally represents that there's potential to do work if you were to remove the membrane. There is potential energy stored across this membrane. The difference in charge is potential energy. If we can harness that difference in charge, we can do work. And by far the best example is when we resynthesize ATP in the mitochondria. We love to leverage this idea. We leverage membrane potential to do work in the mitochondria. That's coming. You've got 2 years before you learn that. So if I could measure with a small microelectrode the inside of the cell versus the outside, there would be a negative charge inside and a less negative charge outside to the tune of around minus 70 millivolts. That is, the inside is 70 millivolts lower or has 70 millivolts less charge than the outside. And this is typical, it's average, but it moves. And that movement is the basis for graded potentials and action potentials. But it also moves normally with those leak channels, always letting some sodium and potassium cross. Now one cell doesn't often move between minus 40 and minus 90 just on its own. There are different types of cells in the body that keep different resting potentials. Cardiac cells, for instance, pacemaker cells keep a more negative potential and lose that potential more easily versus some other cells in the body. So the types of resting potentials you'll see if you survey the cells in the body are between 40 and 90. And here we see our candidates. Factors that cause the membrane potential. We have number 1, unequal distribution of leak channels. More potassium than sodium, allowing more potassium out. Number 2, we see that the anions can't move. They're relatively large. They are these big red balls down here that are proteins. And then these light blue or teal balls are larger phosphate ions. They're too big to slip out of the ion channels, even though it looks like this cartoon would allow that. They are too big to move. And then 3, we see the sodium potassium ATPase working non-stop using energy. ATP is broken down to pump 3 sodium out and 2 potassium in. Now, we took longer than I thought discussing this information. And what I really want to do is jump way ahead and show you this video before we leave. We should just have enough time to fit it in, because this will help pull together what we've talked about and what we're going to discuss on Tuesday next week. This won't be in the recorded YouTube video because the sound doesn't work. I need to play it over the speakers. So we're going to watch this and then we're done.